All right, so I do biology by the ton. I think a lot of people get that feeling too, where for a long time people looked at individual genes and spent their sort of entire career at that, in that one site. But as whole genome sequencing has gotten pretty cheap, we've expanded that. We're now hunting the genome for every possible instance of our feature. And so we end up looking at many, many, many sites. And so to me, visualization is important, not because I can sort of dig in and look at exactly what's going on, but I need to look at thousands of things to try to figure out what I should even start looking at. So that's why my talk is about rapid structural variant visualization. We're going to look at different sequencing technologies, how to visualize all of those together, and then how to go through and really pick out what structural variants you might want to further investigate. All right, so this is a little awkward given Diana's case against a linear reference, but we'll stick with linear reference model for now. All right, so probably many people know about sequencing, but fewer people might know about structural variation, so we're going to kind of start at the beginning. All right, so with Illumina paired-end sequencing, we take the entire genome, we shear it into small fragments, and then do a size selection, which is a pretty important aspect of structural variant identification. So all of these sequences roughly come from this distribution. In this case, it's about 500 bases on average. Then each of these sequences goes onto a flow cell where there's a sticky end. That sequence then sticks there. It bends, forming this little bridge. We then replicate this sequence. We release the two ends. And for each of these fragments, we now have a reverse strand copy and a forward strand copy sitting on this flow cell. And this happens billions of times on a flow cell. We take some nucleotides with little light bulbs on them. We wash them over these sequences. And when they bind, they flash, and we take a little picture. And from that little picture, we can start to decode the two ends of our fragments. So we take, so a little shortcut here. I'm going to go ahead and just give, since this is the reverse strand, I'm going to go ahead and decode that and give the reverse complement base. All right, so then we take more nucleotides with more light bulbs, and we start washing them over these fragments, and we end up with what we call paired-end sequencing. So we have a single fragment, and we actually read out the ends of it. So when we do this for all of our fragments, we end up with something that looks like this. We have this unsequenced space in the middle, and then we have actual sequence on the ends of these things. But they come in this jumbled order, and since we don't really know where they come from, this is where our linear reference comes in handy. We take these sequences, and we start looking for where they align. Oh, there was a nice little animation. There it goes. They start looking for where these sequenced ends align to the reference, and in the end, we get something that looks like this. All right, so what can we determine from this alignment? Well, first we can see that there's this little gap here where there don't appear to be any sequences landing in this space. We can characterize, this goes really fast, I'm not going to use the door. So we can characterize the overall coverage, that is the number of alignments there are at every position in this blue line here, and you can see that there's this stretch where there are no alignments. So this is the first type of evidence that we have that something strange is going on in this sample. We can also look at the characteristics of every pair. So we have a lot of pairs that look quite short, and then we have a few pairs that look quite long. We can actually characterize how unexpected the length of an individual fragment is if we go back and look at that expected size distribution of our fragments. So some of them are exactly what we expect, and some of them are much larger than we expect. We also have these sequences where the actual sequenced end is not continuously aligned. The first bit aligns to the left, and then there's a big gap where the end of it aligns. We call those split reads. So given that distribution and looking for the split reads, we can go ahead and characterize all of the pairs that we've sequenced. You add in the coverage track, and now we have some evidence of a structural variation in our sample. So this G and this T are only next to each other in the sample, but not in our reference. Right? So that's the gist of paired-end sequencing and identifying structural variants in that sequence technology. We also have long-read sequences from places like Oxford Nanopore and PacBio. These work a lot like split reads, where you have these continuously aligned sequences, and then you have these gaps in the alignment. And these gaps are evidence of a structural variant. A little bit newer technology is 10x. This is slightly complicated. It starts with a long molecule, and then isolates that molecule, fragments it, and then sticks a little barcode. In this case, we'll just say all of these are blue. And then it does Illumina paired-end sequencing on those fragments. But that barcode gives us this information that all of these paired ends came from the same long molecule. So when we go back and we align these sequences, they're Illumina paired-end sequences, we get not only the distant paired-end evidence, but we can also get this read cloud evidence, where we have all of these molecules, all of these pairs came from the same molecule, but there's this big gap in where these different alignments cluster. All right? So we have paired-end sequencing, we have long read sequencing, and we have linked read sequencing. And it all seems pretty straightforward how to identify a structural variant from these types of sequences. But like a lot of the things that Deanna was talking about, things aren't so simple when we look at less complex regions of the genome. So in this sample, we have this CA, that's got the other end, which is GAG. But what if this GAG occurs many, many places in the genome? When I go back to do my alignment, I have many valid alignments. One of them is totally normal, and several of them are evidence of a structural variant. And the way that our short-read aligners work is if there is a repetitive sequence where some fragment can align equally well to each of these, the aligner picks one of them randomly and sets that as the aligned position. So you're actually likely to get this type of alignment from this type of sequencing. So the end result is we are still quite bad at detecting structural variants, especially when we compare how good we are for detecting small variants like SNPs and indels. All right, so here's a pretty recent result from a project called Deep Variant from Google. They used machine learning to detect SNPs. And here is their results. So there's this, on the y-axis you have precision, on the x-axis you have recall. In case you've forgotten, precision is basically of all of your calls, how many of them are true positives. Recall is how many total events are you able to detect. And we'll talk about an F1 score, which combines precision and recall. All right, so here's their result. They actually do make an improvement. I think the hilarious to me thing is the x-axis goes from 0 to 100. The y-axis goes from 99 to 100. So if you can't see what's going on, I'll zoom in. Actually, we need to zoom in a little bit more to really get a sense of the improvement here from machine learning. I'll remind you that we have precision versus recall, and these big bars were labeled. That's 100 and 99.875 on the x-axis, 87 and 100. And then using my Photoshop skills, I'm able to estimate with varying precision here that this one is at 99.9545455, and the deep learning gets us an improvement of the F1 score by a little tiny bump. And if you're wondering what it takes to get into the Atlantic, it's that kind of improvement. So if you want to get written up, all you need to do is have a pretty marginal improvement in sensitivity here. All right, so I'm glad this is being recorded. All right, so this is where we are with SNP calling. All right, this is state of the art. Every machine that Google has and all of their impressive machine learning gets us this kind of improvement so we're basically at perfect, right? F1 score of 1. All right, so where are we with structural variants? This is a really recent paper that looked at almost every structural variant caller that exists. So those are all along the x-axis. And the y-axis is this F measurement. I think the importance of this graph is twofold. One is a lesson of when never, never, never to use a stacked bar chart. Stacked bar charts are quite interesting, but I want you to tell me on average, how well do we detect duplications? I have no idea, right? So anyway, but I think you can get the sense that there are 4 different types of variants we're looking at the F1 score for. So at best, I'm guessing here, the maximum value would be 4, so you get F1 of 1 for all 4 techniques, and we are far from that, right? So we are doing, especially when we compare to SNP calling, we're doing quite poorly with structural variants. So these are all short reads. You might think long reads make things better. they've gotten rid of their stacked bar charts and used a little bit better staggered bar chart, but you can still see, even with long reads, even in simulated data, we still don't have great results, especially with respect to SNP calling. So what do we do if we want really good calls for structural variants? We use IGV, right? We've already seen a couple of slides with the integrated graph genome viewer, probably what that's called. So I think right now I have to go ahead and say that IGV is great. All right, and if we look at a section, so this is a known structural variant. IGV, you can see the coverage plot in gray up top, and you can kind of see that it dips there in the middle, then jumps back up, and then you see all of those alignments that I showed you before. And IGV is great if you really want to dig in to a specific area. You can zoom, you can scan, you can change colors, which is great. The problem is you have to zoom, you have to scan, you have to change colors. If I really, the ideal view for looking at structural variants requires many clicks. You have to click here, right click there, you have to, okay, well, this is paired in data, so I first want to see all these as pairs, okay. Oh, I start to see some discordant pairs there, but if I want to actually see how many there are, I need to go back in, do some more clicks, sort by insert size, and okay, now I can see that there's paired end evidence and there's coverage evidence that the structural variant exists. You have to do this at every single site. I'm sure some of you know exactly what I'm talking about. Looking at even a few dozen variants in IGV gets incredibly painful. What's even worse is that almost all of your data is on some remote server somewhere else. It's 130 gigabytes, and you want to view these things in IGV. So what do? You take little tiny slices of your BAM file, you download those. That doesn't really work. You try to do the remote viewer. That doesn't really work. You sort of give up. You look at half a dozen of these structural variants, and then you move on. So this is the main problem that I was dealing with when trying to figure out high-quality structural variants, and it was the motivation for creating samplot. It's an open source tool. I'll be happy to take money from it, but I don't require it. And what we wanted here is we wanted a very, very simple command line program that took minimal arguments and in about 1 second gave you, so this is the exact same view of the exact same loci from the same sample, gives you a PNG in just a couple of seconds. So this is quite nice because I have 3,000 plus structural variants I want to look at. I don't want to spend an enormous amount of time creating the data and downloading the data. Now the size of those PNGs is a tiny fraction of the total BAM size and also much faster. So let me walk you through this plot really quickly. So along the x-axis you have genomic position. This top bar corresponds to the structural variant query. So you see up there I had samplot, then I gave the chromosome, the start, and the end, and the type. So that is where that bar tells me where am I looking. And then everything under that bar is the raw data from the BAM file. Samplot makes no sort of judgment whether or not a paired end evidence or the coverage at all corresponds to your query. It's up to you to make that decision. But in here we are giving paired end reads, we also give split reads. And the insight that we had is we want to generate this image in 1 second, and we want you to spend 1 second looking at it, and that's it. We want you to decide immediately, is there enough evidence, yes or no? And so what we did is the y-axis for these paired ends corresponds to the distance between the aligned ends, or the insert size. So you remember way back when I showed you that expected insert size distribution, if there's a deletion between two aligned pairs, the distance between those is going to be much larger than if nothing's going on. So along the bottom here, you have all of the normal alignments, and along the top, you have the alignments that support the structural variant. So you can see both from and then we also here in gray, we give the per base coverage. That is how many reads align to that exact base. So you immediately see two types of evidence that support your structural variant. You see the drop-in coverage underneath that bar, and then you also see pairs that clearly support the existence of that variant. And then you also see this is a heterozygous variant. You see normal alignments along the bottom through that variant, and the coverage doesn't drop to 0. So you've learned a lot about this variant doing no clicking whatsoever and spending 1 second on it. All right, so that's the gist of samplot. I want to spend as little time as possible analyzing this data. But there's a lot of features that we've built in. As we heard a few times already, there's other information that we know about the genome in each place. So we allow for extra annotations underneath your samples. And there's a little bit of intelligence here. So the first annotation track here is the common SNPs. So this is, the format here is a simple bed file. If you provide samplot a bed file that's a chromosome and a start and an end, it will draw a little rectangle spanning that position, and that's it. But you might have extra information. So if we're looking at line elements, I actually have names associated with each of those intervals. So if your fourth column has a string in it, samplot will take that string and put that above your bar. You can use this for repetitive elements or genes or other types of annotations you might have in the genome. And then the third one is, it might be a little bit hard to see here, but the depth of the color of those lines can be adjusted if you have a float value in your fourth column. So in this case, we're looking at mappability, so basically how unique, how well a sequence might align there is corresponding to how unique the sequence is. So 0 means that it's a perfectly unique sequence, and then as you get darker, or as this float value gets higher, the mappability goes down. So you can see that this variant almost certainly spans some sort of repetitive element. Or obviously, yeah, it's a line 1, right? So there's this correlation between the repetitive element and the uniqueness track. And then we also, the last track, if you just have a GFF file that has information about genes and transcripts, that can also be annotated below. All right, so we have samples, we have a lot of extra genomic information, but really when you're looking at rare disease patients, you're almost always looking at trios and you're trying to figure out, is this an inherited variant or is this a de novo variant? So samplot also supports multiple samples. So here we have a daughter up top and then we have her parents below. So tell me, is this an inherited variant? Tell usually corresponds to a verbal. Is this inherited? What kind of, so let's say sample 8, 9. Does anybody, who knows NA12878? Is her father 8, 9 or is her mother 8, 9? I've forgotten, I should know this. Yeah, so their blood is actually, I did my postdoc at Utah and their blood is actually stored in a freezer just to the left of my desk. But so let's say the father is 8, 9 and the mother's 9, 9, 0. So what version, who gave, so is she heterozygous or homozygous, the top individual? Heterozygous, right? So who gave her the copy that had the deletion? 8, 9, right? 8, 9 is homozygous, so 8, 9 has 2 bad copies. 9, 0 has 1 bad copy, 1 good copy. She's heterozygous, so clearly her mother gave her the copy without the deletion, and her father gave her the copy with the deletion because he had no choice, right? And we spent almost no time, and we know the exact, not just the genotype, but the inheritance pattern of this variant in this individual. And this is exactly what we want with samplot. We want you to look at an image and throw it away and never look at it again. All right, so we've looked at multiple samples. We've looked at extra genome annotations. Samplot also supports different types of sequencing technologies. So in this case, we're looking at Oxford Nanopore. So those are very, very long sequences. So we don't see any pairs here, but we see in orange is the aligned section, and then the dotted lines are the gaps that exist in an alignment. So this little dot here connects a sequence that existed in the individual. It's then aligned. That alignment has 2 aligned sections and 1 large gap, which is evidence of a deletion. We also support 10x. So 10x, remember, takes a long molecule, fragments it up, barcodes it, then does Illumina paired-end sequencing. So you see pairs, you see split reads, and what you also see is you see 3 tracks here. So what's going on? What 10x does with that long-range information, it actually assigns every pair, or at least tries to one of 2 haplotypes, the mother haplotype or the father haplotype. We'll say mother is the top one, father is the middle one, and then they can't assign every single pair a haplotype, and if they can't figure it out, they have this undefined. So we're not just looking at a collection of alignments, we're looking at alignments per parental genome. And so now, again, you can see this is the same variant in the same position. You can see exactly who contributed this variant, which haplotype had the variant, and which haplotype did not have the variant. The extra information here is this thin green line connects all of the fragments that were in the same original long molecule. It's called a gem for 10x, so they take one long molecule. That molecule, all of the contributing pairs from that molecule are connected by this green line. So you can kind of trace the actual event or non-event across the region. All right, so looking at multiple technologies, looking at multiple samples, we can actually do all of this at once. So in many cases, we're sequencing individuals with both long reads and short reads. And in case that's a fact for you, you can look at all of that data together. So here we have two types of Illumina paired in sequencing on top, followed by PacBio, then Oxford Nanopore, then 10x sequencing. All of the long molecule sequencings have haplotype resolved pairs, so you'll see three different plots for each one of them. And you can see they're all basically in agreement with, depending on the types of evidence, we see more or less, or the depth, you see more or less evidence. But I think the picture is quite clear here of what's going on. All right? All right, so this is great, right? I've just given you a way of generating thousands of images for every structural variant you possibly want, and you can quickly download those and flip through those. Well, I mean, that's a huge pain in the ass in itself. You still have all of these images. How are you possibly going to track, oh, this one is good, this one is bad? You end up with an Excel spreadsheet, and life is horrible. So what we did is we put together a software package called svplotIt, and this takes advantage of the AWS cloud to manage that curation process. So we take a bunch of structural variants in a VCF, you take all of the alignment files or the BAM files, and you generate all of those images with samplot. However you want. If you want multiple samples, if you want multiple annotations, whatever you want, you put the configuration in there. You generate several thousand files. We're actually doing this with salmon. I don't know if anybody knows anything about the salmon genome, but it's jacked. And so we have like 100,000 variants we're trying to look at. And it takes all of those images, uploads them to S3, and then uses DynamoDB to actually track your opinion. So you vote, you get a web page that looks like this, and you're asked the question, is this variant real, yes or no? And you're going to answer yes, or you're going to answer no, and your response becomes a row in DynamoDB, which is a pretty basic database that Amazon supports. And the nice thing about this structure is you can have teams of any size. Amazon will scale to however many people, whether it's one or 100,000 individuals, and you can be spread across the entire world. There does not need to be a central server running all of this. All right, so this one, I already gave the answer. We would say no. And to make this very, very fast, you don't have to click on it. We've done key bindings. So you can click on no, or you can hit the N on your keyboard, so you can just tear through these things. So we look at another variant. What do you think? Yes or no? Yes. I agree. So that becomes another line. And then once you've gone through all of your variants, you go up here and you can look at a report, and you actually get the percent of individuals that said yes for a variant, or the percent that said no, or I don't know. And then you can use this to help curate your overall structural variant call set. All right, so we did a little experiment. We had 1,300 structural variants. We had 9 researchers, which were basically postdocs and graduate students in Aaron Quinlan's lab. And we spent only 1 hour looking at all of these variants. And so here's the time spent. On average, it took these individuals, and most of them are not structural variant experts at all, but it took on average 2 seconds. So this is double. I was hoping people would spend 1 second, but I think some people got up to go to the bathroom, obviously. But on average, you can just tear through these variants and annotate. The grad student in the salmon project, she looked at 50,000 variants herself in just a few days, which I think is roughly as awful as pipetting things into 96 well plates, but I've never done that, so I'm not sure how awful that is. All right, so is the annotation, is manual curation useful? All right, so this is a little bit of a complex plot. Every circle is a variant. And there's different tools out there that can give information about the variant. One of those is called SVTyper. So you give a query variant, and it tells you back the genotype. So in this case, SVTyper, if the SVTyper returned that the individual does not have the variant, then they are referenced, that circle is blue. If SVTyper said the individual is either heterozygous or homozygous alternate, then they're red. So every circle is a variant, and the color indicates the genotype from SVTyper. The y-axis comes from a copy number calling tool called CNVnator. So if you give CNVnator a region, it compares flanking coverages to your query coverage and will estimate, is there 0 copies, that is, that you've lost both of them? Is there 1 copy, which means you've lost one of them? Or are there 2 copies there, which means it's normal? All right, so we have multiple views, and then along the x-axis here, we have the mean curation score. So we did 9 researchers. Everyone said yes or no. Yes means that variant got a 1. No means that variant got a 0. Across those 9 individuals, we took the average score, and that's the mean curation score. So so all of these were unanimously good. All of these were unanimously bad. So in general, you can see there's a lot of agreement between the 3 methods. But you can see, looking at that green dot, or sorry, the what color? Blue. The blue cluster, I do need a pointer. The blue cluster right here, so mean curation score of 1. SVTyper is calling all of these as reference variants, and CNVnator is saying they are at copy 1, meaning that they are non-reference, they're deleted. So SVTyper clearly has some false negative issues. And then you also see up here that SVTyper said that they're, wait, hold on. Yeah, then there's also issues where the copy numbers are off because we said that clearly there does not exist a variant here, but we see that there appears to be copy number 1 from CNVnator. So we actually can control for some of the weaknesses that exist for how we judge whether or not a structural variant is real or not. And in fact, the 2 seconds that you spend actually looking at the variant is typically less time than SVTyper or CNVnator spends running their models on that data. All right, so let's try it out. Who got the email? Okay, so it should look like this. A very conspicuous, thinking that some foreign state is trying to capture your data. That might be true, but it's also from me. All right, so click on the email, and it should look like this. But wait, don't do anything until I explain this. This is not the most logical series of steps. So important pieces of information here. 1, the email that was sent to you. So in this case, ryan.layer@gmail.com. This is case sensitive. I'm sorry. I know you have multiple email addresses. I sent it to just one, and I have a specific combination of capital letters and lowercase letters. You need to use that exactly. So that's the first piece of information. The second is your temporary password down here at the bottom. Your second piece of information that you must. So your exact email address and this number. All right? So now everyone that has done these 2 things. So now please click on this link. All right, so this should take you to the main login page. Don't do anything. First thing, click this button. You're confirming a new account. Okay, now put in your case-sensitive email and your temporary PIN. Make sure you've checked this confirming new account. All right, and then hit submit. The first thing it'll ask you is please change your password. We don't have any password requirements here. Make it as insecure as you'd like. Or as secure as you'd like. Sort of pessimistic. All right, so once you've done that, now submit your changed password, and you're ready to go. All right, what you should do, who's gotten this far? Okay, so now click on Score Images. You will be presented these images in a random order. There's like 53. Not like 53, they're exactly 53. Click on that and you'll be given your first image. And so on this one, what do y'all think? Yes, No or I don't know? No. Okay, that's all the training you need. That's all the training that we gave people in the experiment. I don't want to bias you as to what you think is a good and bad, but please, yeah, your turn. All right, and I'm going to, so one of the nice things about SV Typer is that you can, of just showing, I had to hand copy all of those emails. Minimize that. So I can actually pull down your results and run any sort of analysis I'd like on them. So I'm going to kind of do that on the fly here. Let's do this. So there's 50 images, 2 seconds per image. It should take you no longer than 3 minutes. Don't worry, I have your email address. I can tell exactly how slow you're going. So I'm totally willing to shame anybody that's going too slow. All right, so how's it going? All right, so what I can do is since all of that information is up in DynamoDB, if you have the right permissions, you can just at any point grab all of those query results. So is this big enough? I'll do a little bit bigger. So I have this command here. It's this retrieval.py, and I give it this configuration file that was generated when I made the project. I hit enter. Let's count how many rows. So we have we already have 886 annotations. Anybody done? Nice, nice. I asked the Hackathon committee for budget for Frisbees, and they said no. So if you're going to do ask you how you liked things, say I'm very disappointed I did not get a Frisbee. All right, let's do this again. Oh, we've got 20 more, so things are progressing. Who's still going? Peter. He does structural variants. What's going on? All right, so for those of you that are done, which is, I guess, Peter, we can talk about this later. So what you get for every variant, you get the email address. So if you trust people, or if you don't trust people, you can ignore them or not ignore them. The next, you get the actual S3 path of the image that they curated. Then you get their, so that's this long string here. You get their answer, and then you get some metadata about the variant. Its chromosome, its start, its end, and we record how long they spent between their last one and their current one. So if you're really pressing your graduate students, you can say, you're just not performing, I'm sorry. But, okay, so we have all of this data, and then we can do a quick little Python hack. I've even forgot what sorry. Fake JSON file, parse, that's a good name. So if we parse that, so basically what I'm going to do is, so this data comes from 1,000 genomes, phase 3. So in that data set, they have identified variants as being reference, heterozygous, or homozygous alternate. That is, you've lost a copy, you've lost 2 copies, or you've lost no copies. So I'm going to compare what the genotype was to what the curation was. So, ooh, that didn't work. All right, so up here, then let me do some, since this is a bioinformatics, I felt like I should do some sort unique-c, count sort dash. Okay, so what's going on? Anybody? So I'm taking all the results, I'm sorting them, doing unique-c will tell you how many unique ones are all in a row. That'll give you a number plus that unique string, and I'm sorting that. This is a little tutorial too on how to be awful command line people. All right, so here's our so far. I'm pretty impressed how fast we went. So how do we do? We had some I don't knows, which I'm disappointed in all of y'all. That's almost, we had what, 90, nearly 90 of those. But okay, so All right, so 0 copy loss. That means it was identified as heterozygous. This is pretty good. So there's 618 0 copy loss. That means, and you said no. So this means you agree with that genotype. There's also these up here where there's a little bit of I don't knows, and some people say yes here, which there are errors in 1,000 genomes data, so this is not gospel. But yeah, so you get a sense of how your genotyping compares to how your curation scores go. And so you might think, oh, I trust that there's nothing going on here, and I trust, so single copy loss, you said yes. 2 copy loss, you said yes. So I might trust that the genotypers and my curation both agree here. Maybe these are my high quality variants. All right, and I think that you've all done a really great job. I'm very proud of you. Give yourself a round of applause. Peter, did you finish? Peter finished, great. Question? This one? Well, so I think that there are cases where the genotypers fail. We showed that in the results, right? There are false positives and false negatives in using either copy number base to give you genotype or looking at pairs, which is what SV typer does. So in those cases, you might think of your manual curation is cleaning up or can clean up some of those issues. So I would consider these, when there's differing views, that's sort of ambiguous. Maybe go look at those. But really, you can completely ignore 618 things, and you can probably just trust 204 things as being, yeah, we don't need to look at those again. Biology by the ton, right? I look at as few things in detail as possible, right? Okay, so thank you all very much. I think that actually went better than I thought it would. All right, so I think that, for those of you that got the email and have computers, you're probably pretty confident when you look at an image in very quick time, you can say, is it 0 copy, is it reference, 1 copy loss, or 2 copy loss? It's pretty straightforward. You look for differences in coverage, you look for pairs that might confirm the variant. Well, this made us think, well, if people can do it, maybe machines, right? Obviously, every talk has to have machine learning in it. Deanna left. She didn't talk about machine learning at all. Yeah, so we took so Murad in my lab took the TensorFlow library, which is deep learning, yeah? And we trained, right? So we took 1,000 genomes, which the trivia question is how many genomes are in 1,000 genomes? Obviously 2,504. So they did SV calling on that fairly recently. And then what we did is for every structural variant, we sampled up to 5 images of 0, 1, and 2 copy losses. And that gave us 94,000 images. You see the distribution here is roughly what you might expect. They're mostly, most variants are rare. Actually, most variants are singletons in this data set. So you have 65% 0 copy loss down to just a few 2 copy losses. We took those 94,000 images and we split them into 2 sets, 74,000 to train on and 18,000 to test on. Training with a GPU only took about a half an hour. And we trained up to 93% confidence on those held out 18,000 images. So, okay, so I always ask when machine learning, like, what now, right? I've got 93% accuracy. That helps nobody, right? Well, what we did is we actually saved the model, which is about 4 megabytes. And now that we have this model saved, for every new image, we can use this model and classify for a given image, we actually get this probability distribution. So the likelihood that it's a 0 loss, a 1 loss, or a 2 loss. So given a new set of images, I now get this little vector, and from this vector, I can see what does the model predict the copy loss is. So it actually does very well. We're in like the 98 to 99% agreement between 1,000 genotypes and this model. So they did better than y'all did. I'm sorry, as far as agreements. So you see here the top one, the highest value 0.999. Corresponds to no copy loss, which I think we can all agree that's the state there. And then you have heterozygote loss, and you have homozygous alternate, which is 2 copy loss in the bottom one. All right, so this is really quite exciting. All these images, and it takes a millisecond or so to take an image, take our model, and get a prediction back out. But it doesn't always work. Okay, so when does it go wrong? So this top one was predicted to be with pretty low confidence, predicted to be heterozygous, but in fact it is clearly zero copy loss. So what's going on there? Anybody want to guess what the machine was confused about? That top one in the back? Yeah, so you're saying like right, it might be looking right there? Yeah, so I think there's a couple things. You have a big drop here in coverage, and you also have a bunch of junk along the top. And remember, in a lot of these heterozygous cases, you have these normal alignments are on the bottom, and the discordant alignments are along the top, right? And so my guess is, and this is one of the things we're trying to not just guess about, actually pull it out of the model. I'm guessing that it got thrown off, like you're saying, about the coverage is a little funky, and for some reason, these reads are very high. All right, so in the second case, the called genotype was reference, but the model predicted there was actually a 2-copy loss. So what do you think it was keying on here? Feel free to answer. Yeah, the blank space. Yeah, there's this big drop in coverage here, right? So maybe that there could be a gap. Maybe there is definitely an event going on. So we need to make things a little bit better and have the query variant correspond to the event. We're still working on that. We actually cropped out everything. When we trained, we crop out everything, all these words, all the axes, the bar up here, so that to hopefully make the training a bit better. I mean, it's quite good, but maybe we can do something to make sure that the event matches the query. And then this bottom one, you actually see an incredibly confident reference, but 1,000 genomes genotyped it as being heterozygous. So what do you think is going on here, right? This counts against our accuracy, but I actually think we are doing a better job predicting the state than whatever was used to genotype in 1,000 genomes. All right? Anybody, what do y'all think? Do you think this is reference or heterozygous? It's kind of weird. If anything, there appears to be a slight increase in coverage underneath this deletion. All right, so beyond doing machine learning, we're also doing a lot of new visualizations. We have this thing I call split view. So one of the big problems we have in very large structural variants, so here we have a half a megabase deletion. That is a bunch of the genome to go through and actually plot. It takes a lot of memory and it takes a lot of time. And really, all the action is occurring in this tiny fraction of the overall space. So what we implemented is a way to basically zoom in. So now we specify a boundary around the variant, around the ends of the variant, and we only plot, so we're only plotting a small fraction of the space here. You can see a little ellipsis there and a white bar there to show you, all of this is from over there and all of this is from over there. And it's also 4 times faster. So you're actually seeing where the action's happening, and you can do these things much faster. The file sizes are lower, and it takes a lot less RAM, which is quite helpful when you're doing 100,000 of these. Another really nice thing about split views is it gives us a way to look at translocations. So we did an epilepsy project where we identified a de novo inverted translocation in an individual with a de novo inverted translocation. And that, in this case, we guessed that it disrupted this GABRE gene, which this is a family of genes that are known to be related to epilepsy. And as Maria pointed out, in IGV, you could never look at this. But here, you have chromosome 2 is all of this space, chromosome X all over this space, and you have this line. Blue indicates that it's actually an inversion, and they perfectly span these 2 chromosomes, so you have this inverted translocation. I also, sort of in real time, took a little picture of Maria's HERT2 variant and zoomed in. And so this is her first. So I will say these are short reads because on their repo they did not have the index for their long reads, and that would take me several hours to download and several hours to generate the index. So this is the short reads. But you see, this is her first translocation from 17.8, and this is her second translocation from 17.8. So I did this because you can do these samplot images on remote files. As long as they're on FTP sites or HTTP sites, you don't need to even have like direct access to those BAMs or need to download those BAMs. You just point at the URLs and you're able to, in just a few seconds, generate these images. All right, so we've got a lot of stuff. One of the things that was really missing was we have a way of having teams of 1,000 curate these structural variants. There's a little bit of overhead on AWS setup, but if you're going to have a lot of people and a lot of variants, it's totally worth it. But most of us are just working by ourselves, have 1 or 2 or 3 BAMs, and a set of variants. And I want to look at those, and I do not feel like messing with AWS. So we actually now have this really neat, if you want to check out this tiny URL as a small sample, given a VCF, you do samplot_VCF on it, and it generates the images and generates this little web page locally that you can just click on individual variants and you see the samplot image below. All right, so I don't know, I think I'm pretty good on time. Yeah, so that's it. Samplot is totally open source. My GitHub site, I got to thank Murad did all of the machine learning stuff. We started this project back when I was at Utah. So John, Joe, and Brent contributed a lot and continue to contribute a lot to this. And yeah, these are my boys, Chip and Jack, and I'll take any questions.